Welcome to this European webinar series on microglia. I'm Guy Brown from the University of Cambridge, and my co-host today is Jake Dundee. This is a series consisting of monthly talks of about 45 minutes by European scientists talking about their research on microglia, followed by a Q&A session. And today's talk is by Agnès Nadjar from the University of Bordeaux in France, and the talk is entitled "The Effects of Nutrients on Microglial Function." So during the talk, please write your questions in the chat function as the speaker speaks, and direct the questions to all. Please keep your microphones muted and cameras off to prevent feedback and distractions and preserve bandwidth. So I'll now hand over to my co-host Jake, who's going to introduce the speaker. Jake.
Thank you very much, Guy. So Agnès did her PhD in Bordeaux, Professor Dancer, where she discovered the field of neuroimmunology. She then went on to work with Dr. Bazar as a postdoctoral fellow to work on the role of microglia and neuroinflammation in Parkinson's disease. Then in 2009, she moved to Boston to work with Professor Hayden, and there she studied the interactions between microglia and astrocytes in establishing sickness behavior. When Agnès returned to Bordeaux in 2011, she obtained a permanent position as an associate professor. It was during this time that she became interested in the role of nutrients in microglial function, and focusing specifically on the postnatal period. Then in 2009, she obtained a faculty position, moved to another institute still in Bordeaux to develop this new theme, focusing on the role of microglial bioenergetics in development of obesity. And with that, take it away, Agnès.
Thank you very much, Jake, for this introduction. First of all, I'd like to say that I feel really honored to be part of this webinar series on microglia, of which I think I haven't missed a single issue since its creation. So thank you, Guy, Jake, and Claire, who was the co-host before you, Jake, for organizing this for all of us. So today, I will try to convince you that nutrition is a potent modulator of microglial function at all stages of life, with a special emphasis on the role of lipid nutrients on the phenotype and function of these cells. So, So we all know here that microglial cells are highly versatile cells in the sense that they can shift from one phenotype to another, depending on their local microenvironment. And the ultimate goal of this is to maintain the homeostasis of the central nervous system. And among the signals that make microglia shifting from one phenotype to another, they are actually the intervention of local cues like neuronal activity, for instance, or the presence of debris or dead cells in the environment of microglial cells, but also more distal cues coming from the body, like pathogenic patterns or hormones. And all these signals will be computed by microglial cells, and this will modify the way they interact with neurons, so that neurons can adapt their activity to cope with the situation in presence and preserve the integrity of the organism. And we and others have hypothesized that nutrients can also be potent modulators of microglial phenotype for different reasons, including the fact that they serve as energy substrates for the cell. They can also contribute to tissue architecture, including the architecture of the brain. They regulate signaling pathways through their derivatives. And so today I'm going to show you some of the data that we gathered on the role of some dietary lipids on microglial activity, first during the neurodevelopmental period, and I'll show you how we demonstrated that these lipids can control the shaping of hippocampal networks in the developing brain, but also their role in the modulation of microglia oligodendrocytes interaction in the developing white matter. All this work has been recently published or is currently in press. And in the last part of my talk, I will present you a new piece of research on the role of nutrients that we find in obesogenic diet, on the role of microglia, but this time in adults, and all this is still unpublished. So why do we think that lipids can control microglial function? Well, to understand this, we need to go back to basic cell biology. We know that microglial cells are morphologically extremely dynamic cells, and so they need a membrane that is able to go for extreme deformation to allow phagocytosis, for instance, or to allow the motility of microglial processes. But not only this role in the morphological adaptation of microglia, but the membrane is also the site where microglial cells will recognize neighboring cells or neighboring patterns. And this happens via the expression and anchoring of specific receptors in the membrane. So for all these reasons, we think that the lipid composition of microglial cells can be key in controlling their function. And the most abundant lipids that are found in the cell membranes are the polyunsaturated fatty acids, or PUFAs, that you may know better under the terminology omega-3 and omega-6. And these lipids actually constitute one of the two hydrophobic tails within the phospholipids. And because of their degree of unsaturation, these lipids can control membrane fluidity and flexibility, but they can also control the conformation of receptors within the cell membrane. And another important feature of these lipids that I will talk about a bit later is that they can be released from these phospholipids, so unesterified from these phospholipids, and then they become precursors of bioactive lipid mediators. So the brain is particularly rich in PUFAs, and the main PUFAs that are found there, so for the omega-3 family, which we are also called N3 PUFAs. So during the talk, you're gonna see N3 PUFA or omega-3, but these are synonymous. So the main omega-3, it's the DHA, and DHA is also known for its anti-inflammatory activity. And the main omega-6 in the brain are the arachidonic acid, or AA, and the DPA-N6. And these lipid species are precursors of pro-inflammatory mediators. An important characteristic of these lipids is that they have to be obtained from the diet. At least the precursors have to come from the diet, because our body cannot synthesize them. But once we ingest these precursors, then we can produce all the derivatives, so all the long chains that will then reach the brain. Another important feature of the PUFAs is that they will be massively incorporated, specifically during the neurodevelopmental period, from the last week of gestation till the end of lactation in the rodents. So for all these reasons, we were wondering whether modulating the amount of PUFAs in the diet of the mother could actually affect the microglial phenotype and function in the brain of, in the developing brain of the offspring. So this work has been initiated by Charlotte Mador when she was a PhD student in the lab, and now she's back to Bordeaux and she's developing her own group. And it was also conducted by Quentin Lérol, who was also a PhD student at the time, and two postdocs, Lydie Morel and Moira Rossito. And what they did is that they exposed mothers to either a diet that was sufficient in omega-3, sufficient in the sense that we were giving the proper amount, the proper ratio of omega-3 on omega-6, as recommended by the World Health Organization, which is of 1 omega-3 for 4 omega-6. So the control group, they had the good amount of omega-3 in the diet. And another group was fed with a diet that was deficient in omega-3. So for these mice, we didn't provide any precursors of the omega-3. And this led to an omega-3 deficiency in the mother and in the pups. And this is closer to what is observed nowadays in the Occidental diet that is known to be deficient in omega-3. And so the pregnant dams, they were exposed to this diet from the first day of gestation till the end of the lactation period. And at P21, we studied the hippocampus of the pups. So why P21? Well, because this is at that age that most of the main maturation processes are over in the hippocampus. And also because this is the end of this massive accretion phase of PUFAs in the brain. And why the hippocampus? Because in this project, we were particularly interested in the effect of these dietary manipulations on the cognition of the animals. And first, we wanted to test whether modifying the maternal diet could actually impact the lipid composition of microglial membranes in the offspring. So to test this, we sorted microglial cells from the brain of P21 pups, and we ran some lipidomics analysis to study the lipid composition of microglial membranes. And we found that when the animals were developing under an omega-3 deficient diet, we could see a significant reduction in the amount of the main omega-3 in the brain, the DHA, which was correlated with an increase in the amount of the omega-6 DPA-N6. So the fact that the animals were developing in a context of low omega-3 indeed was modifying the amount of PUFAs in the membrane of microglial cells in the sense of a decrease of the omega-3 and an increase in omega-6. And so then the obvious question was, what is the impact of these lipid modulations on the phenotype of microglial cells? So for this, we collaborated with Oleg Butovsky at Harvard, and we did some transcriptomic analysis of the homeostatic signature of the microglial cells. So this homeostatic signature, actually, it's the study of the expression of a set of genes that is known to, when these genes are expressed all together, this is the marker of microglia in a physiological state. In our conditions, we found that omega-3 deficient microglia had an alteration of this signature with the upregulation or downregulation of at least 1/3 of this gene set. And when looking more closely to the type of clusters of genes that were modulated by the diet, we found that there was an alteration in the genes involved in the homeostatic signature, of course, but also fatty acid metabolism, which was not a big surprise since we were modulating the amount of lipids in the diet. And we also found an increase in the expression of genes involved in the immune response and inflammation. And this is about that time that Rosa Paulicelli and Beth Stevens have shown that during development, microglial cells will be essential for the refinement of synapses and spines in different brain areas, including the hippocampus. And this happens via an increase of their phagocytic activity towards the spines. So in our conditions, we wanted to have a more functional assay to see if the lipidomic and transcriptomic modulations that we observed before had a functional impact on the cells. And so we tested in a very global manner the phagocytic activity of microglial cells ex vivo. So we sorted microglial cells from the brain of the P21 pups, and we exposed them to pH photobioparticles that have the capacity to fluoresce when they are in an acidic compartment, like a phagosome, for instance. So the more phagocytic the microglial cells, the higher the amount of fluorescence will be emitted by the cells. And this is what we quantified by FACS. And you can see that at all time points of the kinetics, omega-3 deficient microglia were always significantly more phagocytic than the omega-3 sufficient microglial cells. But then we wanted to see if this had some relevance in situ. And so for this, we teamed up with Marie-Ève Tremblay in Canada, and she did some electron microscopy on IBA1 stained microglial cells. And so what you can see here, you can see nicely stained microglial processes in the CA1 region of the hippocampus. And you see here, for instance, the processes that is opposed to a synapse. So you see the terminal and the spine. And she could also observe spine inclusions within microglial processes. And she quantified these events. And she found that in omega-3 deficient microglial cells, there was a significant increase in the number of contacts between microglial cells and the synaptic clefts, and also an increase in the number of spine inclusions within microglia. We confirmed this data by performing double immunofluorescence for IBA1 and PSD95, which is a marker of the postsynapse. And we could confirm that there was an increase in the colocalization between the two signals in omega-3 deficient animals. So it seemed that microglial cells in omega-3 deficient mice display an increased phagocytosis towards spines. We wanted to check whether this had an actual impact on the morphology of neurons in the hippocampus of the animals. And for this, we did some Golgi staining analysis. As you can see here, we quantified the number of spines on the dendrites of the pyramidal neurons in the CA1 region. And we could find that indeed this exacerbation of spine phagocytosis was correlated with the significant reduction in the spine density in omega-3 deficient mice. However, with such an approach that is modulation of the diet, of course, we will have a very broad impact on all the cells of the brain. And so we were wondering whether this increase in the phagocytosis of spines by microglia, is it driven by the fact that microglia are modulated in terms of lipid content, or is it synapse driven, or is it both? So to test this, we sorted microglial cells and synaptosomes from the brain of omega-3 sufficient mice and omega-3 deficient mice. We exposed again the synaptosomes to pH photo. We coupled them to pH photos so that we were able to follow by fax the phagocytosis activity of microglial cells. And then we made all the combination possible. So for instance, we exposed omega-3 sufficient microglial cells to omega-3 sufficient synaptosomes or to omega-3 deficient synaptosomes, and the other way around. And of course, we did this from pre-21 pups brains. And then we did some fax analysis to quantify the phagocytic activity of microglial cells in all these conditions. And here are the data. So I'm going to take you through this graph slowly. So we observed that when microglial cells were sufficient in omega-3, so this bar and that one, we couldn't see any effect on the phagocytic activity of microglial cells when the lipid status of the synaptosomes was modified. So when microglial cells were sufficient in omega-3, whether the synaptosomes were sufficient or deficient in omega-3 made no difference on the phagocytic capacity of the cells. However, as soon as the microglial cells were deficient in omega-3, there we always observed a significant increase in the phagocytic activity of the cells. And here again, no effect of the lipid status of synaptosomes. So this data suggests that this is the microglial lipid composition that is the main driver of spine phagocytosis exacerbation in our condition. And for the sake of time, sorry, I'm not showing you the data, but we also showed that this increase in spine phagocytosis was actually due to an overexpression of the complement receptor 3 protein at the surface of the microglial cells. But then we really wondered how the fact that we were modulated the membrane lipid composition of microglial cells could actually affect their capacity to phagocytose spines. And so this is what we deciphered in the rest of the study. And to understand the logic behind, you have to remember what I told you before, that these omega-6 and omega-3 that are contained within the cell membrane, they can be cleaved out of the membrane, and then they become precursors for hundreds of lipid derivatives. And these lipids can then control signaling pathways, they can control the expression of transcription factors, and so they can ultimately and indirectly control gene expression. So they can have a very wide effect on the cell biology. And so we wanted to draw a map of the lipid derivatives landscape in these microglial cells that were either deficient in omega-3 or sufficient in omega-3. And here, again, we did some lipidomics analysis, and we found that omega-3 deficient microglial cells had a higher amount of lipid derivatives that were coming from omega-6 precursors, as you can see here in blue, while the omega-3 sufficient microglial cells were containing more lipid derivatives coming from the omega-3 precursors, here in orange and purple. But when we looked more closely to the actual data, we found that only 8 of these species were significantly modulated by the diet. We found that 4 omega-6 derived lipids were significantly increased in the cytoplasm of microglial cells, and 4 omega-3 derived lipids were significantly decreased in these cells. So then we wondered whether one of these lipids or the combination of several of them could actually explain this increase in the phagocytic activity of microglial cells. So to address this, we first used an in vitro setup, so primary cultures of microglial cells, and we exposed these cells to these lipid derivatives that I just mentioned before in presence of pH photosynaptosomes. And we followed the accumulation of fluorescence for 3 hours, as you will see here. So you see the microglial cells in culture, and you see this increase in red fluorescence, which is a marker of the increase in the phagocytic activity of the cells toward the synaptosomes. And when we quantified these data, we observed that only when we applied this lipid, the 12-hit, only this lipid derivative was able to significantly increase the phagocytic activity of microglial cells. And this lipid is an arachidonic acid-derived mediator and is produced from AA by the activity of this enzyme, the 12-lipoxygenase or 12-LOX. And so in the next study, we tested whether this had some relevance in vivo. So is it because in vivo microglial cells are producing more 12 hits that they are more phagocytic towards the spine in the hippocampus? So to address this, we sorted microglial cells from the brain of the pups, again at P21, from the brain of omega-3 deficient mice in this case, and we exposed the cells to bicalene, which is an inhibitor of the 12-LOX. So we were reducing the production of 12-hit in these cells, and we observed that this was sufficient to decrease the phagocytic activity of microglial cells ex vivo. And so with all these data and other data set that I'm not showing you here, overall, what we showed is that when the maternal diet is low in omega-3, there's a profound modification of the microglial lipid metabolism with the activation of this pathway, the 12-hit pathway. And what we were also able to show in this paper is that this lipid derivative was responsible for the increase in the expression of CR3, which then leads to the hyperphagocytosis of the spines. But then we wanted to see if this modification of the microglia neuron interactions in the developing hippocampus could actually affect the behavior of the offspring. So to test this, we used the YMS task, which is a way to study the spatial memory performances of the animals. And for those who wouldn't be familiar with the YMAs, during the learning phase, we put the animal in the starting arm. And then the animals are allowed to visit the 2 arms of the maze for 10 minutes, and the third one is closed by a door. After 10 minutes of exploration, the animals are removed from the maze, put back in their home cage, and then 1 hour later, we put them back in the starting arm. But this time, they are allowed to visit the 3 arms of the maze. So because the mice are extremely curious, if they remember what happened 1 hour before, so if they have good memory abilities, they should spend more time in the novel arm as compared to the familiar one. And this is exactly what we quantified in the animals. And as you can see here, the omega-3 sufficient mice, they were perfectly able to recognize the novel arm from the familiar one. So they were spending more time in that arm than in the familiar one. However, omega-3 deficient mice, as you can see here, they were spending the very same amount of time in the 2 arms, meaning that they were just choosing the direction by chance, randomly. So they had deficits in their spatial working memory. And we know from the literature that actually in this YMS task, when the animals are at the choice point, so when they have to decide which way to go, when they are doing the right choice, there will be an increase in the synchronization between the hippocampus and the prefrontal cortex. And this hypersynchronization of the two structures is absolutely essential for the mouse to make the right choice. If we don't see this hypersynchronization at that very moment, at the choice point, then the animals will choose randomly the direction to take. So we wanted to see whether in the omega-3 deficient animals, because they had these spatial memory deficits, if this was due to a decrease in the connectivity between the 2 brain structures. So for this, Cantalerol collaborated with Cyril Desjean in Bordeaux, and so they did in vivo double electrophysiological recordings by placing probes in the hippocampus and in the prefrontal cortex of the animals, and they studied the synchronization in the activity between these 2 structures. As I told you before, so when the animals are doing the correct choice, when you look at the activity of the neurons when they were at the choice, in the choice zone, you should see an increase in the coherence between the 2 signals. So you should see, and this is represented here as a heat map, so you should see an increase in the synchronization of the activity of the hippocampus and the PFC. However, when the choice is incorrect, this time you shouldn't see this increase in synchronization between the hippocampal neurons and PFC neurons. And actually, this is exactly what we observed in omega-3 deficient mice, where we saw a decrease in the synchronization between these two structures. And this was perfectly correlated with their poor memory performances in the task. So omega-3 deficiency leads to a decreased synchronization between brain structures, which may be due to a decrease in the myelination of the white matter tract, which is one of the important factors in explaining the connectivity between brain structures. And this is about that time that this exciting literature on microglia oligodendrocytes interactions started to pop up. And so from all the papers that were published at that time, we now know that microglial cells invade the white matter very early during the postnatal days. And they observed these fountains of microglia, a term what actually was originally coined by Kirschmann in 1939. And once these microglial cells are on site in the white matter, there were release trophic factors to promote oligodendrocyte proliferation and maturation, factors like IGF-1, for instance. But not only this, but microglial cells will also promote myelination of the white matter by phagocyting pieces of myelin. And it may be counterintuitive, but actually this is essential that microglial cells phagocyte myelin to promote myelin turnover and to promote the myelination of the brain tract. So we were wondering whether omega-3 deficiency could actually affect microglia oligodendrocyte interactions during the brain development and what would be the consequences on brain myelination. And this work was performed by Fanny Decker, who was PhD students, and Quentin Leroy. So the first experiment that we did was to quantify the number of microglial cells that were invading the white matter of the animals that were either sufficient in omega-3 or deficient in omega-3. And we looked at different time points during the postnatal period from P0 to P15. So according to the literature, we should see an increase in the number of microglial cells at P10. And this is exactly what we found in the omega-3 sufficient animals. However, as you can see, in the omega-3 deficient mice, we saw a significant decrease in the number of microglial cells around that time, which suggests that there was a lack of a decrease in the microglial invasion of the white matter in these conditions. And here we studied the corpus callosum in this study. So then we decided to look more closely to microglia axon myelin interactions at P10. So when we saw this differential number of microglial cells in the corpus callosum of the animals. And for this, we did electron microscopy experiments again in collaboration with Mariev. And as you can see here, so here you see a microglial cell in the white matter and an axon with the myelin that is very easy to recognize with the different layers of the myelin sheath. And we quantified the number of contacts between microglial cells and myelinated axons in this white matter, but also the number of myelin debris within microglia, since I told you before that this phagocytic activity of microglial cells toward myelin is essential for proper myelination of the brain. And we observed that in omega-3 deficient animals, there was a significant decrease in the number of contact between microglial cells here in blue and myelinated axon here in pink. But there was also a decrease in the number of myelin debris, as you can see here in green. So there was a decrease in this number of debris in microglial cells. And so it seemed that in the developing white matter, there's an alteration in microglia myelin interactions. So the next question was, does this have an impact on brain myelination during development? So first, what we did to address this, we quantify what is called the G ratio. So the G ratio allows to evaluate the thickness of the myelin sheath because it's the division of the diameter of the axon on the diameter of the axon plus the thickness of the myelin sheath. So the higher this ratio, the thinner the myelin sheath. And this is exactly what we observed in omega-3 deficient mice, where we found a significant increase in G ratio. And this was particularly true for axons of a smaller diameter. So the myelin is thinner in omega-3 deficient mice. And then to have a broader analysis of the integrity of the myelin tracts, we used DTI MRI imaging, which allows to quantify the diffusivity of water molecules in the tissue. And in a tissue like in the white matter that is extremely well organized with the myelin tracts that are very close to each other, actually water molecules shouldn't be able to diffuse too much in all the directions of the space. And this is what we quantified. We quantified this diffusivity coefficient. And we observed that in omega-3 deficient mice, there was a significant increase in this coefficient, meaning that the water molecules were able to diffuse more, which actually was a marker of a decrease in the integrity of the white matter in omega-3 deficient mice. So as the first intermediate conclusions of all this work, I hope I was able to convince you that first, maternal nutrition can affect microglial phenotype and function in pups. Also that more specifically, the amount of omega-3 in the diet can actually, and especially when this amount is too low, this can alter the lipid composition of microglial membrane and their lipid metabolism. And this will ultimately modify microglial neuron interactions and lead to a misshaping of neural networks in these animals. We also, in this work, we were also able to describe a new pathway that is involved in microglial phagocytosis, which is the 12-LOX12-HIT pathway. And we showed that the activation of this pathway could enhance spine phagocytosis. And actually, this could have relevance for some neuropathologies because it was shown that it's in some mouse models of AD, this pathway can be activated at the periphery in macrophages. So the question is still open as to whether this pathway could also be activated in microglial cells in the brain of these animals, and why not explain the fact that these mice have lower amounts of spines on their neurons. And not only the microglial cells from the gray matter were affected by the diet, but also white matter microglia displayed alterations, which led to alterations of oligodendrocyte physiology and myelination processes. And altogether, all these alterations lead to behavioral deficits in the offspring. And with that, I'm gonna finish by thanking all the people who did this work, and especially the first authors on this paper and the people who did all the work. So you can see them highlighted in orange here. I will also thank all the collaborators. So Richard Bazinet for all the lipidomics analysis, Marie-Ève Tremblay, with whom we did the electron microscopy experiments, Oleg Butovsky for the transcriptomic mapping of the microglial cells, Pierre Gressens, with whom we co-supervised the PhD thesis of Quentin Lérol on all this work on myelination in the omega-3 deficient animals, Cyril Desjean for the in vivo electrophysiology recordings, and finally Arnaud Mourier for all this work on mitochondria and the bioenergetics analysis. Of course, I will thank also all the funders, and I wanted to tell you that there are PhD positions available in the team, so if you are looking for one, do not hesitate to apply. And I will thank you now for your attention.